All right. Welcome back from tea, everybody, to session 2. Apologies to our Zoom participants to have to make you wait. If you knew how cold it was here and how nice the sunshine is outside with a warm cup of tea, you'd understand. This session focuses on biodiversity and conservation, which has become one of the cornerstones of the GSSA's topics and interests. And I'm just waiting for my first speaker to come in from tea. She's right here. So our first speaker is Anisha Diaram, who is one of the key people involved in our national vegetation map, which I hope you all know it and have perused it and enjoyed it. It is an incredible product. I think it must be the envy of all other countries with interesting vegetation. And what is so exciting is that it's a living project. And well, you know how complex our vegetation is here in South Africa, but it is constantly being updated, and she will give us the heads up on the latest updates.
She was doing so well, she could have probably done my talk for me. But thanks for the introduction. Yeah, so I'm Anisha. I work with the National Vegetation Map, and it's a project that's been going now for, think about 17, 18, yeah, 2000, yeah, for a long time. And so I think if who, by show of hands, who uses or has used the vegetation map at some point? Awesome. Cool. And I'll convert the rest of you hopefully today. So basically what we do is over the past 10 years, we've been updating the map, and we've had 4 versions. And now today, what I'm going to take you through is a very quick overview of what's in the 5th version, which we just released. So it's the it's the 2024 version. We will release a beta version. We'll leave that up online for all of you to use for a while. We get feedback, and then we release the final version. So what's in there, which is what I'm going to take you through, is we've made updates. We do this in an iterative process. So over the course of the years, different parts of the map have been updated. And the data comes from our community of practice. So it's all of you. And in this last iteration, what we've had is a complete new update. Oops, wrong one. Complete new update of the whole of the Northwest Province. Some new forest patches in the Langeberg. Small changes in Elam, Fehlerkriet, Fynbos. We've had 5 new Strandveld types along our southern coast. Some of those used to be dune thickets, but they are now Strandveld types that we've put in with the Fynbos. So that was work by Richard Cowling and Adrian and team. And then we had my mistake, fixed a little whoopsie in the fish music thicket. And then we added some mangrove information, and this is really cool. What we're trying to do is we're trying to include in the map, you'll know mangroves are very dynamic, but often we talk about losing ecosystems. But with mangroves, because of some of the structures that we've put in place in our estuaries, mangroves have actually increased. So what we've tried to do is we've tried to capture the historical extent and then also the contemporary extent. But I'll show you what I'm talking about a bit later. So in terms of the classification, we've had quite a few new types added. There's 7 of them. 5 of them are from those new Strandveld types. And then the 3 of them are coming from that northwest change. So they're mostly savanna types. And because of the changes along the Strandveld, the new Strandveld types basically overlapped with where we used to have something called Blondbos Strandveld. So that's no longer in the veg map. It's been updated with these more refined classification. And so what does this mean for the grasslands? So in this iteration, if you look at the area in red, they've been all just changes to the extents of the grassy types in those areas. Those are the types that have been affected, and it's just boundary shifts. And they're in the eastern part of the Northwest province. So that's what you can look forward to when you look in the map. And then further along other parts of the map which you may be interested in, changes you may encounter, is we had previously in 2018 included estuarine habitats, so things like seagrasses and a few other very fine scale information within the estuarine area. But what we found is that the estuaries team from NMU was updating the data and the map was getting left behind. So what we rather say is go to their database if you want that data, because you don't want to cause confusion. So that's no longer in the map. You'll just see the big estuarine area, no habitats in it. And here's the mangrove information. So we had a lot of help from the estuary community, and they reviewed their data, they sent us information, and we tried to make sure that our information matches their delineation. So what we went to is old imagery, but then also these really useful and really cool references. And what they have is little mappings of mangroves. So you'll see that, is this St. Lucia? There was a berm put in across the whole estuary, and so there used to be just tiny patches of mangroves, but now there's this massive stand that's grown in. So we're not saying in the veg map whether that's good or bad. We're just saying here's where it was, here's where it is, and then my colleague Mapale, when she does the red list assessment, that's her problem to decide with her community of practice what that actually means. But what you'll find in the veg map is this new field called the epoch mangrove, and you'll see classes for contemporary, which is where it is now, a class for historical, where it used to be, and then another class for where it has been persistent, so it will have both fields in there. So that's really cool. We'd like to do it for more ecosystem types if we have the data. So if you know of information, please let us know. And then when you click the link, so I'm not sure the announcement might have made its way to some of you. I think I'll send it to the Grassland Secretariat, and then we can send it to everybody who's interested. When you click the link, it's currently living on a what do you call these things? Google Drive. So if you use the Biodiversity Advice or BGIS website, you'll know that it's under construction a little bit at the moment. That's one of the reasons, but also because with the beta version, it makes it easier for us to correct errors. So if you go to that drive and that link, you'll find that there's the spatial layers. So there's a spatial layer that you can use with QGIS and ArcMap or any other software. There's also a KMZ if you don't have that software and you just use Google Earth. And then there's also, like we've had in the past, CariMap, so the mobile version. But what's really cool is with the update of CariMap, we now added some of the descriptions of the veg type. So it's not just the name, you can also see more or less what you should be looking at. And on the drive here, we were using it. It was quite fun to see. We could recognize things that the description was saying. So that's quite cool. There's also this time we put in colors for ARC and for QGIS. So if you use QGIS, you'll see the real veg map colors. There's a folder with the description of the new types, and then all the technical reports. So all the changes I mentioned, there's a technical report associated with it that tells you who was involved, which references we used, and exactly what the changes were. But what I want to say is, if there are formats that you would like to use, like maybe one of the things I want to add is a geopackage for QGIS users. If there are other things or other ways that you think the veg map would be more useful, just let us know and we can adapt, and we're always happy. We want you to use it. So anything that helps you use it, just let us know and we'll do our best to cater to that. So those are the changes if you find errors in this version. But what's really, really important is we're always looking to source any new areas for the next version of the veg map. So if there's an area that's really irritating you about the veg map, gosh, these people just get it wrong all the time. It's basically, I'm inviting you to complain. So please complain to us. We don't know what's going on all across the country. If you know of an area, just let us know, and then we can work on a proposal for that. Right. So over the years, this is just a quick overview of, oh, whoopsie. We've had the 2006, which was the first version, and you can see much of the map hasn't been changed. But over our various versions, we've managed to get almost 25% of the area of the map updated. We'd like to refine more parts of the 2006 map. We know that the map's not perfect. It's still useful, but we want to make it more useful. So again, if you have areas that you want to work on, that you know we can do better, please let us know. If we look at the distribution across the biomes, for example, in 2018, we had a whole reworking of the thicket biome. It's been different across the different biomes. For the grassland, we managed to update about 20% of the map, just over. So we still have a long way to go if there are areas to change. And this is just by how much of the area of the map has been updated. But what we also would like to work on, and we started doing that with students, with Professor Van Aert at UFS and some professors at FITS, is we've got students working on refining our understanding of the description of the veg type. So what actually makes up the veg type? Those short descriptions in the veg map book, it's not enough to tell us the richness of what occurs in a particular grassland. So if you have that data, if you have students working on that data, if you have a reference, please let us know so that we can then add that information. We're now curating the descriptions as a database, so it doesn't have to be a book. It's in this, we can create a mega spreadsheet, and then we can add that information so we have that a better understanding of our ecosystem types. Then we had quite a few discussions this morning about talking to the global community about our ecosystems. And some of the work that SANBI has been doing, which is very exciting, that some parts of the global community have started coming to us and saying, do you have information? Can you please plug in your data into these international processes? And the IUCN community has been working quite closely with SANBI because South Africa, like Susie said, South Africa stands out in terms of the way our data is organized, is often ready to plug into some of these systems. And so one of the things that you may have come across is that they've been building this ecosystem typology. So it's an IUCN global typology. It's not perfect. There's lots of global typologies. This is one of them. But what's useful about it is it feeds into things like the red list assessment. And they have a hierarchical structure which sits at a level higher than our ecosystem types, so vegetation types. Our vegetation type sits somewhere here, so it gets aggregated up. And what they've asked us to do is try and fit the South African ecosystem types into one of their classes. And you'll see there's this table that we've been working on. It's a small team at SANBI. We've asked some experts to help us. And there's ecosystem types that don't fit perfectly. A lot of ours fit under this temperate grassland, but you'll see we don't think it's a perfect fit. It also fits under some other of their groupings. But this is a first pass. We've told them this is a draft. We need our community of practice and our experts to please help us review this and refine this. So here's an opportunity. It's not going to fix everything, but it's a chance for, if you have a moment and you'd like to help us, please let me know, and you can help us review this table and give feedback and say, ooh, you got it really wrong here. Please, can you, we think that it rather fits within this part of their system, or it's a terrible fit, nothing explains this grassland type. So yeah, it's an invitation to please help us so that the data that we send to them is a better fit. And we're able to communicate our ecosystems in a much better way. And one of the places and the reasons that this goes, of course, it goes to the IUCN database, but there's a second global process that's related that will also piggyback on the IUCN group's work. There's a global ecosystem atlas project. So what they are doing is they are trying to collate a huge atlas of all the maps of ecosystem types that's available from around the world. And what's really nice is, again, they came to South Africa and they said, can we please have your ecosystem types, but you give it to us from your perspective? And do you have any other African partner countries that also have ecosystem maps of ecosystem types? And so we've been working with them and letting them know of the ones that we know of. So that's really nice. It's our opportunity to say, this is what we have from our perspective. This is what our ecosystems should look like. And that IUCN work, they're using the global typology and as a higher level, but it points back to the original data set. So it will point back to our data set. So having and refining that information in our veg map makes a massive difference, because it then feeds into these global systems. And so it's more of a bottom-up approach from us. At least that's the hope. I think there's probably still a long way to go, but at least they're starting to recognize that they should be speaking to us to get our data. And that's it. So if you have anything that's on your mind, if you want to chat about the veg map, if you want to assist with helping refine that crosswalk with their global typology, please let me know. We really, really value your input and your help. Thanks.
Thank you so much, Anisha. Your time is up in 2 seconds. So we'll save the questions for Anisha to the general discussion later. And so I'd like to invite up our next speaker, Peter Mashaba, who's going to talk about riparian vegetation and how it has changed along the Olifants River.
Thanks. Hi everyone. My name is Peter Mashaba. I'm from Nepal Wilderness. Studied at UT for a master's. So I'm here to present to you the project we did in the Olifants River system, together with Prof. Willem Mebek and Dr. Panagos. So we look at spatial temporal changes in the riparian vegetation of the Olifants River systems over 25 years. So riparian systems, as we know, there's a lot of terminology used to describe these systems. So they're ranging from riparian zones, riparian areas, riparian ecosystems, as well as riparian vegetation. So this study only focused on riparian vegetation. So definition of camper, which is what's common in South Africa, applies in this study, which basically is vegetation that is in close proximity to rivers, and then it is defined as a riparian zone. So it's normally dependent on the river for a number of systems and functional traits, whereby it also displays structural, compositional, and functional differences to your adjacent terrestrial vegetation. It is also basically a vegetation that is distributed based on inundation and the gradient of the riparian bank. So this vegetation is normally influenced by geomorphology as well as your hydrology, normally being your floods, your rainfall, and as well as geology and sometimes elevation. So, and then it differs again in composition from adjacent systems like the terrestrial vegetation, but sometimes some biomes, like in the grassland biome, that is not the case. So you'll find a riparian system, terrestrial and the riparian bank have similar vegetation. So this influence manifest across different spatial and temporal scales. And there's also dimensions where these systems happens. It's basically your longitudinal from the source to the mouth of the river, as well as the vertical underground movement of water, and lateral from your edge and to your higher lying bank. So back in 2000, Mybeck published his PhD, whereby they looked at he identified 17 plant communities across this system. So the communities were basically in the grassland and savanna biome, and then 8 and 9 communities were classified respectively. So the phytosocial study of Mybeck provided an opportunity for us to really look at spatial and temporal changes of the plant vegetation over 25-year period at the plant community level. So this study aimed at to investigate spatial temporal changes in the plant vegetation of the Elephant River over 25 years, and also looking at to evaluate changes in plant communities associated with both the biomes, and also look at disturbances that occur within those two biomes. So basically looking at to find out whether the riparian composition and cover change over this period, and also are there any differences in the distribution and cover of alien and invasive plants. So this is the area where the study took place, basically from Brighton, where the river originates, and just downstream of the Fleck-Wishelo Dam in the Sekhukhune land. So as I mentioned, the study was stratified back in 95 with the aim of identifying, describing the plant communities of the Elephants River. So baseline data from 1995 was basically used with the data from data collected in 2020 and 2021 to really compare spatial temporal changes. Those plots were resurveyed in 2020 and 2021. So basically in the grasslands, there were 26 sampling plots from 13 sites, as well as 35 sampling plots in the savanna biome from 18 sites. So only a subset of the actual sampling plots were surveyed in this study. So basically, this is just all the sampling plots and the representative communities that were resurveyed in 2020 and 2021. Infill procedure just involved the modified urban bronchial approach, whereby we used a minimum variable bulk transit of 200 square meters. So both riverbanks were surveyed. It means at one sampling site, we surveyed both riverbanks, and the length and width of the sampling plot was basically determined by the width of the riparian vegetation, as well as the floristic parameters in that system. So all written plant were recorded, also including the composition of indigenous and invasive plants. The growth forms were assigned, as well as canopy cover was assigned using the plant number scale. So for the sociological approach was used in this study, so whereby we firstly classified the data of 2020 and 2021 with the one back in 1995 for us to be able to determine any changes within species composition. We also statistically we use K-Quest test because the data is from account data that we used to assign cover values, so it was not normally distributed. So we use chi-square to test for differences. So now on the grass. So in the grassland biome, as you can see, there is a there is an upward trend of species richness from upstream to downstream in the grassland biome section. As you can see, species richness increase basically from 150 species to about 200 species over 25 years. So that's a huge jump of species richness that is coming into the system. So it is only a plant community for that only showed a significant increase in richness. Biggest contributors were your forbs and your gramineas, basically the grasses, your sages, that contributed to the highest species richness. There's also notable increase of woody plants in the system, so where it varies from a single woody to almost 11 species that came in new into the systems. Alien plants also made a contribution into the systems, whereby 18 were alien plants, and invasive contributed about 12 species of the total species richness over 25 years. Canopy cover follows similar trends, but there were special variations whereby the upstream communities increased in cover, and then the midstream one declined in cover. So again, here only plant community 4 had a slight decline in cover in the process, and then it went on towards plant community 4, whereby it increased slightly in community 7. The herbaceous growth form again were attributed to this increase, basically your gramineae and your forbs, as well as some of the woody component that came in the upstream, which boosted the cover of the whole communities up. So this is can we cover trends per growth forms per communities. As you can see in the upstream communities, plant community 1, 2, and 3, there's a bit of increase in the herbaceous layer, your gramineae and forbs, and they increased significantly. But as we start moving downstream, their cover starts going down, and then there's manifestation of woodies that are coming in from plant community 3, and then we go down to your plant community 6 and 4 as well. There's a slight decline of woody cover in the downstream plant communities 7 of the Grassland biome, but this is mainly due to certain species that were controlled, which I'm gonna speak in the invasive component part. So this is species composition changes that manifested the community. So here I'm going to highlight your dominant and species that are strongly competing in the community. So you can see over 25 years, there were changes in composition, whereby some species were really reduced in cover, and then as other species came in. So for example, in community 1, Ergotis plana was relatively replaced by Cynodon dactylon in the 60 as a strongly competing species. This shows that across all those communities, you can see in 1995, basically there are new species that are coming into the system that were not really prominent back in 1995. So it shows over 25 years, the Grassland biome had species compositional changes over time. So this is further down to plant communities and plant community 7, whereby again, compositional changes are being shown along these communities. Your gramineae, you have Hematheria artesium plan and Ariantha that were basically over 25 years replaced by Separus marginatus, Separus fasciatus, and Paspalum digitum. So all of these compositional changes occurred in different communities over time. In the Grassland biome, similar trends were observed of species richness, whereby species richness increased longitudinally across the system. So overall, species richness increased from 237 species to 341 species. So that's quite a huge increase in richness in the system. Basically, it ranged from 20 to 87 species that came in, basically in plant community 2 and community 4. So similarly, again, in the savanna biome, your herbaceous layer plays a huge role in species richness over time. But interestingly so is that your invasive species contributed 7.2% of your richness in 1995 and increased by about 2% over 25 years to 9.6. Again, similar trends, again, when I speak with total cover, same trends were observed where cover increased slightly, but plant community 2 has a big jump in cover. So again, plant community 2 is basically in mountainous terrain. So all of your gramineae again came in hugely and then contributed about 16% increase of cover that you are seeing from 39 to 55%. As we go downstream, we saw that again, as the river goes downstream, cover declined a bit. So plant community 4 had a decline in cover as well in the Grassland biome. So what we have seen again is that in plant community 4, all communities displayed an increase in canopy cover of both indigenous and invasive trees. The trends of the growth form also show that the woody layer in the grassland was more stable, as we can see in community 1, 2, as well as community 4. But in community 3, there was variation where some trees declined and then woodies, and then some of the forbs came, some of the shrubs came in. Composition analysis as well showed similar trends whereby some species that increased in cover, like your acacia delbata increased in cover, but other indigenous species changed over 25 years in different communities and peak growth forms. As you can see, the differences highlighted in this slides. Your forbs, only 2 were dominant and strongly competing, but over 25 years, other species came in and were strongly competing in 2020. So in the discussion, I'm looking at, so in the grassland biome, your forbs surpassed your gramineous as in terms of richness over 25 years. So furthermore, we saw an increase in woody species in the grassland biome over 25 years. So the economic cover trends are variable in the grassland biome, whereby there was an increase in the upstream and then usually a decline in the downstream areas. So the grassland biome is associated with different land uses, like from your mining, agriculture, both commercial and communal land, residential areas. So all of these land uses could be a factor in contributing to these changes. For instance, your normal decreaser species declined in the grassland biome and they were replaced mostly by your robust increaser 1 species. As well as in Plant Community 7, the decline in cover was mainly due to the decline in Acacia delbata, which was basically controlled and then boosted your indigenous species to come in. River regulation plays a big role as well in these systems, whereby when during periods of low rainfall, dams are controlled and then they release basically low flow. So this also, when they open the sluice gates, those kind of large excess water erodes riverbanks following periods of dry areas. I also like to emphasize that even though these systems are compromised by both compromised by both natural and human disturbances, as the river increases in size, the drainage increases and the scoring effect of the water increases, whereby we see the decline in the downstream areas. So this is the control of plants that are happening and the influence of fire in the system. In the savanna biome, similar trends were observed in the system, and an increasing economic cover as well as within community variation occurred in the savanna biome. Similar land use affected the savanna biome as well, whereby all of those edge effect species near the river systems were washed away, and then also due to the side effect of the development, things reduced the cover of your shrubs. But community 2 also, the increasing cover is mainly due to the buffering effect in community 2. Basically, it's mountainous, there's rocky areas there, and the trees are allowed to grow, hence the big increase of cover in those areas as well, in community 2. So lots of them downwards is community 3 and 4, which basically land use is agriculture, citrus farming. So there are a lot of non-plant pollutants coming into the system, increasing the cover of woodies as well. So overall, I can say plant community 4 was changed in structure from the savanna biome changing structure from a thicket to a woodland type of vegetation based on some of the vegetation changes that happen in the savanna biome. So big point is that the system allowed invasive plants, as I mentioned, to come into the system over time. So okay, just in short, the system allowed cultural change in the plant vegetation, whereby over 25 years new species came in. So these changes were basically a manifest of different land use types that manifested into the system. So as the new species that came in, other species came out. So it's a different array of combination of factors that affected the changes in the plant vegetation of the Olifants River. So different land use management approaches are needed at the local scale to ensure that the diversity and richness of the species are maintained, and then also to reduce the bringing in and control of invasive species. Thank you.
Thank you, Peter, and sorry for chasing you off, but we do need to stick to time. Our next speaker is Wataru Tokura, who's going to speak about estimating bare soil fraction in the Namakuru. And while he makes his way here, I just want to remind everybody to please adjudicate the talks. It's very, very easy. Keep coming. You just snap the QR code, and there's a simple little Google form. You've got to give 3 scores. It's very easy, and it really helps us to evaluate. Thank you.
Hi, everyone. I'm Wataru from University of Cape Town. I'm doing PhD for my project. Today, I'll talk about my remote sensing work, estimating fraction bare ground cover in Namakuru. So this map is from a national biodiversity assessment done by Sambi in 2018. And when we look at my area, which is Namakuru here, we see that all the biomes are classified as least concern in terms of an ecosystem condition. But when we really visit it in the field, we see all sorts of different conditions on the field. So we want to know those conditions because these things are important for conservation, agriculture, and policy making. And South Africa has a long history of degradation, but what we need for now is more spatially explicit and a map that we can frequently update. To do so, use of remote sensing can provide a solution for that. But remote sensing has a own set of problems in arid area. But these technical issues now have been overcome. And this is an example in Little Karoo that the study was done by our colleagues. And when we look at the map, so these left, right. Left-hand side map is from Google Earth. And when compared to the remote sensing map, we can see the good correspondence and for the bare patch, which is probably the area suffered for the past overgrazing, can correspond well to the remote sensing product. So the objectives of my project is to mapping severely degraded rangeland using a fraction bare ground cover as a proxy. And to do so, use a similar approach, which was successful in Succulent Karoo. And by doing so, I'm aiming to contribute a biome scale condition mapping. For the method, so the Namakuru was much larger than I thought. It's a huge area, and to get enough training data set is rarely a challenge, but I was very fortunate. So Dr. Dutoy, Justin, he has been taking more than 2,000 photos from different places in Namakuru, and I can use this nice and great archive of his landscape photos as a training data set. So from this more than 2,500 photographs, I selected about 552 photographs based on some criteria for remote sensing work, and read the manually estimate bare ground cover from these photos. To make it consistently, I made a set of reference photos, which was assessed by 3 experts, including myself. So this is a bit of workflow. So geo-tagged photos are the landscape photos that Justin have been taking. And for the satellite data set, I use a Landsat series data set to make a monthly layer from Landsat. And the geotagged photos are split into 30% validation data set and 70% training data. They are put together into a machine learning model, which I will explain a little bit more later. And then model it's validated by validation data set, and then will be compared with field data, and then produce a background map. But for the purpose of this talk, I just started my field data collection, so I'll talk about on the upper part of my slide. So the machine learning model I use is a partial least square regression, which looks a bit scary, but this is essentially a combination of the PCA and linear regression. So my response variable is background read from ground photos, and use optical bands from Landsat series and a bunch of vegetation indices. And because many of these vegetation indices are correlating each other, so put into the single model is a bit of problematic. But by using partially square, it's like making a kind of new things, which I can't explain very well. And then all the data from different years and locations are pulled. But I developed two different models, one for growing season, another one for non-growing season. And then predictor variable was selected based on Q-square value, which is an indicator for predictive performance and also variable importance for projection values. And all the analysis is done in R. So this is the preliminary result that on the left graph is showing the kind of accuracy. So x-axis showing the observation, it's from photos, and y-axis is a prediction. So that model, selected model has 5 bands and 9 vegetation indices. And this dot line is 1-on-1 line, and red line is regression line. And similarly for non-growing season, the model performance is like this. And then I can use this relationship to map across Namakuru. So this is the example for 2023 in December. So it looks good because it's so the dark purplish color is higher bear cover, and yellowish color is more vegetation. So this pattern is corresponding to what we know about Namakuru. So it seems like this model is working. And also if you look at where we are here, it's a Harip Dam, and it's saying that there's no vegetation. So it looks like okay. So when you zoom into some of the places, this is an example for Carnarvon. You can see that gravel road here and dam here, which has lower vegetation cover. And overlay to my map, it seems like this one, my layer, it's working at least okay. And also I can check the kind of accuracy of my model comparing to Justin's photo set. So he has visited this particular site 5 times since 2017. When I compare with my model, so these points are the background estimate from photos. And these lines are the prediction by the model. So the agreement is not 100% perfect, but at least it's following something. In some other area, this example in Bushman's Land looks more accurate and can capture all the recovery after the drought. But unfortunately, there's some places that it has a poor performance like this one. So my model is saying much lower than it should be predicting. So when I map these errors, I can see some points. So these dark red or dark blue points are the points that model says very wrong. So it's some places that still need to have better accuracy in prediction. So the next step, so loss of cover is just a part of degraded rangeland, as everyone knows. And also loss of cover can be from natural causes like pans, or that the area has rock outcrops that it's just a natural law. So we should not confuse with two different things. So the next step is to kind of separate these things together and also adding a bit of different elements to map degraded rangeland. And the lastly, I want to show some of the examples. So these 3 photos are all from Middleburg area. And this red hatched area is degraded land. For example, this one is just right next to town. And that red hatched area is a municipal commonage. And green area is neighboring rangeland. And then the one in the middle is a water point. And the one on the right is an old land. So for this analysis, I use a Sentinel, so the different type of satellite, which has a higher temporal frequency and also better spatial resolution. So when I map that time series of data set, so the green line is the area not degraded, and red lines are showing the degraded area. That's the interesting thing for me here is all those 3 areas, this green line shows a similar pattern. Assuming the pattern of rainfall is somewhat similar because they are all from Middleburg. But in contrary to green line, red line degraded area shows all different patterns because degradation is not just one thing, different kinds of degradation. So that by using this kind of time series analysis, I may be able to pick up some of the features that can be unique to either a degraded area or a good rangeland. The summary, monthly bear grant I predicted for Namakalu, and that approach developed for Succulent Karoo are likely to be working in Namakalu as well. And ground level photos are very important for environment monitoring. And further work needed to validate remote sensing layers and also need to separate naturally bare area. And then I can probably use time series to identify some of the degraded rangeland as well. So my project is part of the SBAP regional project, which is a SAMBI-led project for ecosystem assessment. And I need to say thank you for Justin for allowing me to use his wonderful collection of photographs. Thank you.
Thank you, Wataru, and you're well within time. So we have time for a quick question or two. If there isn't anything straight away, we'll hopefully have some in the discussion session. Thank you so much. Next up is Bridget McMillan, who's going to take us up some altitude to look at environmental drivers of grass species occurrence.
Good morning, everyone. Thank you for introducing me, Susie. So I'm going to tell you guys about the results of some of my master's work, which I was focusing on understanding the factors that influence the species cover and occurrence in the Drakensberg-Maluti mountain range. So plant distributions are influenced by abiotic environmental factors, historical processes, and biological interactions. Each plant species has a specific ecological niche that is caused by these abiotic and biotic factors, which result in a distinct distribution pattern across various scales. This distribution reflects the intricate relationship between a species and its surrounding environment. And understanding these influences is crucial for conservation efforts and for allocating resources to regions of crucial ecosystem functioning. So the spatial variation in species occurrence and cover can offer valuable insights into the ecological dynamics of a system. The factors that influence a species occurrence reflect the necessary conditions for a species survival, while the factors that influence a species cover can reflect the resource availability and disturbances that impact a species abundance. Both species occurrence and cover are complementary metrics. They offer a dual perspective on the vegetation extent and the location of a species. Understanding these metrics can enhance our ability to predict the species distributions and to evaluate how they will change in the future. Species distribution modeling is a powerful tool used to understand a species distribution and to predict how it will change in the future due to climate variation. Fine-scale models can offer an intricate look at the relationship between a species and its model by representing the microhabitat. These can lead to more accurate predictions of the species distribution, which can help with conservation strategies. Abiotic factors like temperature, precipitation, and soil characteristics play crucial roles in the shaping of a species distribution. They can either directly influence a species distribution, or they can act as proxies for other influential variables like stresses, disturbances, and resource availability. Despite the ecological importance of these ecosystems, alpine grasslands are under threat due to climate change and land use patterns. These systems are sensitive to environmental variability, especially the rising temperatures and changing precipitation patterns associated with climate change. Grasses dominate these systems. They play vital roles in water filtration, soil erosion prevention, and are the primary forage for wildlife. Understanding how these species will respond to climate change is important, as they are the key players in the ecosystem functions of these systems. So the aim of my study was to determine what factors are related to the fine-scale cover and occurrence patterns of the dominant grasses in the Maluti-Drakensberg grasslands, and to assess if these factors change with elevation. So, like I said, my study was conducted in the Maluti-Drakensberg mountain range over the course of 5 years, from 2018 to 2023. The study was conducted at 3 locations, ranging from an altitude of 2,000 to 3,000 meters above sea level. The first site was at Golden Gate Highlands National Park at 2,000 meters above sea level. The second site was at Witsi's Hoek, located just below Sentinel Peak, near Puti de Chaba, at 2,500 meters above sea level. And the third site was in the Bokong Nature Reserve in Lesotho at 3,000 meters above sea level. All 3 of these sites are relatively undisturbed by agriculture. However, the Bokong site did occasionally get passing grazing from the goats that were herded through. So for our sampling design, we used a standard grid-based approach where we had an 8 by 20 meter grid subdivided into 160 contiguous 1 square meter cells. At Golden Gate, we sampled 8 survey grids, but at Witsi's Hoek and Bokong, we surveyed 7 grids at each site. In each grid cell, we identified the grass species and recorded their cover. For my study, I'm focusing on the 10 grass species that had the most cover, which made up about 50% of the total grass species. In each cell, we also identified abiotic, and we identified and measured abiotic factors like aspects of mesotopography, rock cover, soil depth, northness, soil moisture, and soil temperature. When I analyzed the data, I used a Pearson's correlation test to test for collinearity, and I selected the predictors that I thought would be the most biologically relevant. I then used a best subset model building approach to generate and identify the best multivariate models for the grass occurrence and cover. I then also evaluated if there was any spatial autocorrelation within these models. And for the models that had significant spatial autocorrelation. I used spatially explicit GE models to account for the spatial dependency. The final models were then selected based on AIC and QIC scores. So the results of my analysis were actually quite shocking, and I'll show you why now. So overall, there was no significant relationship between the species occurrence and cover and any of the predictors. All predictors had most of the predictors had inconsistent effects, meaning that some of them had a positive and some of them had a negative effect depending on the species. Mesotopography and soil depth were the only predictors that occurred in more than 90% of the models, but they also, again, had an inconsistent effect. When looking at the influence of elevation on the predictors, for the highest site Bokong, mesotopography and rock cover were the two predictors that were included in the most models, while for Witsy's Hook, soil depth was the most important predictor, and at Golden Gate, soil depth and soil moisture were important. So because I was looking at the 10 most grass species, you can see that the first 7 occurred at Golden Gate and at Witsy's Hook, while the remainder of the 3 were at Witsy's Hook and Bokong. So because of this variation, it allowed for me to see if there was any variation in the predictor influence across the 3 sites. So mesotopography occurred at all 3 sites, but had an inconsistent effect. Northness occurred at Golden Gate and Witsy's Hook, where it had a positive effect at Golden Gate and negative at Witsy's. Rock cover had an opposite effect, where it had a negative influence at Golden Gate and a positive influence at Witsy's Hook. Now, soil depth also occurred at all 3 sites. However, when the grass species occurred at Golden Gate and Witsy's Hook, there was a positive effect at Golden Gate and a negative effect at Witsy's Hook. But when the grass species only occurred at Witsy's Hook and Bokong, Witsy's Hook had a positive effect, and the soil depth had a positive effect at Witsy's Hook, and it had a negative effect at Bokong. This is opposite for soil moisture. So when the species occurred at Golden Gate and Witsy's Hook, soil moisture had a negative effect at Golden Gate and a positive effect at Witsy's Hook. But when the species occurred at Witsy's Hook and Bokong, it had a positive effect at Bokong and a negative effect at Witsy's Hook. So to summarize, for our high site at Bokong, the most important predictor was mesotopography and rock cover. At our middle site, Witsy's Hook, soil depth was the most important predictor, and at Golden Gate, our lower site, soil depth and soil moisture was important. To summarize the overall effects of the predictors, at Golden Gate, mesotopography and soil depth had a positive effect, while rock cover and soil moisture had a negative effect. At Witsy's Hook, mesotopography and soil depth had a negative effect, while rock cover and soil moisture had a positive effect. And at our highest sites, soil moisture had a positive effect, and soil depth had a negative effect. Okay, so the study highlights that the environmental variables that affect the distribution of grass species are complex and vary with site and altitude. The study also highlights the need for more refined models with more predictors so we can more accurately predict the species distributions. I'd like to thank the University of Pretoria, the NRF, Sandparks, the Lesotho Ministry of Tourism, Witsy's Hook Mountain Lodge, and Transfer Parks Destinations. I'd also like to thank the amazing team that helped collect the data over the past 5 years. We had a great time and experienced lots of wonderful things. Thank you.
Thank you, Bridget. Don't run away. Are there any questions for Bridget? Yes, Lorraine.
Thank you very much. That was very interesting. Just you talked about all these factors and environmental variables and so on, but you never mentioned temperature in any one of those.
Temperature did not come up as significant.
Question and answered. Thank you.
I have a quick question for clarification. You were saying it has a negative effect, it has a positive effect.
Yes.
Was it on abundance and occurrence?
Yes.
And did all species respond the same?
No. So these predictors, they affected the cover and occurrence, but not all species were affected in the same way. So for example, we had Festuca at the highest site and at the middle site. So at the high site, mesotopography increased its occurrence and cover, but at the middle site, mesotopography decreased it. And sometimes if the predictor was important for at the higher site, it wasn't even included in the final models in the middle site.
Cool. Thank you very much. We can pick up on this in the general discussion, but thanks again. Next up is Nell Marie. And she's looking at the rehabilitation of old potato fields. And I genuinely hope she tells us what's happened to the chickens. Somebody know how to play the piano, maybe. Okay, shall we get the next? Okay. Right, apologies for that. We're going to have to wait a bit longer to find out what happened to the chickens. But we will, I hope. But in the meantime, can I call up Mapala Monyeki? And she's going to tell us about the red list ecosystems. Now, I was always familiar with red list plant species, but it turns out whole ecosystems can be in danger too. So welcome, Mapala.
Good afternoon, everyone. My name is Maphala Monyeki, and I will be talking about ways to improve the rate of ecosystem in grassland biome. Okay, for this presentation, I thought that the best place to start will be by highlighting some section of NIMBA that directly speaks to this work and shine light on the mandate of Sunbeam. Having said that, Sunbeam's work is given power through NIMBA, section 52, 1A and B, and section 52.4. So this two section states that the minister and MEC may publish a list of ecosystems that are threatened and in need of protection. And this published list must be reviewed periodically. In fulfilling this legal mandate, SANBI produces a high-level and comprehensive report on South Africa, on a high-level report on South Africa's state of biodiversity every 5 years. And this report basically tells us how we are doing in protecting and conserving our prized biodiversity. So far, 3 iterations of this report called the National Biodiversity Assessment have been completed, and work is underway to publish the 4th NBA report. And its first attempt is represented in the NSBA 2004, which had strong emphasis on special aspect of biodiversity, which included the priority areas and protected areas. And as time went by, data matured and became available, and the scope of NSBA was then expanded to also include broader themes such as climate change and invasive species, which then led to the NBA 2011 and subsequently NBA 2018. As mentioned, work is underway to publish the 4th NBA report. And with each iteration of the NBA, there are advances in terms of the input data that is being used and analysis undertaken, which then improves the quality of our reporting and planning. So the NBA relies on 2 headline indicators, that is the threat status and the protection level. These 2 indicators can be applied to both species and ecosystem across different realms, and they provide a meaningful way of comparing results. So for the threat status, or the red list as globally known, it is based on the IUCN methodology, as mentioned, is applied to both species and ecosystem. And we use this indicator to measure how close our species and ecosystem are to extinction and collapse risk. Just to cycle back, so the first list of ecosystems that are threatened and in need of protection was first published in 2012, if I'm not mistaken. And this list was generated using the national developed set of criteria. And in 2022, August, the second list was then published, and this new list was developed using the IUCN Red List of Ecosystem Standard. So because of the methodology or the criteria used are slightly different, then that means, as well as the input data, like the National Vegetation Map, as Anisha alluded, it has gone significant improvements. That then means that the 2 lists are not comparable. That was just something to note. Okay, now going to, the second indicator being the protection level is a domesticated indicator that is also applied to species and ecosystem, and it allows us to measure how well our species and ecosystem are represented within the protected area network. For the purpose of this presentation, my focus will only be on the Red List of Ecosystem. So as mentioned, the Red List of Ecosystem developed by the IUCN present a global approach of assessing how threatened our ecosystem are. These standards of framework are developed in such a way that they are consistent and can be applied systematically across different realms and are repeatable. So so they were first published in 2016, 2017, and this month, in 2024, the guidelines were updated and the new version is available. So the IUCN List of Ecosystem Guideline consists of three main components. That is the set of criteria, about 5 of them. 2 of those criteria focuses on spatial symptoms of ecosystem collapse, and the other 2 focuses on functional symptoms. And the 5th criteria uses quantitative model to estimate the probability of ecosystem collapse. Overall, they allow us to measure or assess different ways in which an ecosystem may be threatened. We then have a set of quantitative threshold that we use to measure how close our ecosystem are to a collapse state, all right? And lastly, we do have a set of categories that are similar to those, for those that are familiar with the Red List of Threatened Species. So this set of criteria are used to order the risk of ecosystem collapse. So ecosystems that are listed as critically endangered are at an immediate risk of collapse as compared to those that are listed under the least concern. As mentioned, the whole aim is to assess how close or the collapse risk of ecosystem. And the concept of collapse is similar to that of species extinction. And we say that an ecosystem has collapsed once it has lost its defining biotic and abiotic features from all occurrences, such that the characteristics of the native biota can no longer be sustained. Okay, now diving into individual criteria. The first of the special criteria, we have criterion A. This criteria basically focuses on the reduction in geographic distribution of ecosystem or habitat loss, and it consists of 4 sub-criteria. So for an example, criterion A1 focuses on spatial changes that occurred over the past 50 years. And then criterion A2A and A2B focuses on potential future spatial changes over a 50-year period. And then criterion A3 looks at historic declines, okay? And the reason why that they use a 50-year time period is that it is presumed to be adequate enough to diagnose spatial changes with some reasonable level of certainty, while long enough to differentiate between directional change from natural variability. And then A3 uses the baseline date of 1750. This is a baseline date that is accepted globally. It represents an era in which anthropogenic land convention commenced globally. However, that is not cast on stone. Countries or regions that have data, credible data to suggest that land convention commenced prior or post this reference date, they can use that. For South Africa, we are using the 1750 as our reference date. And when the data that is required to apply assessment under this criteria is basically the land cover change as well as the ecosystem map or vegetation map. The second special criteria is criterion B, which looks at ecosystem with limited extent or ecosystem that are narrowly distributed. This criteria uses 2 special metrics, that is extent of occurrence and area of occupancy, to measure or identify ecosystem with limited extent. For an example, the extent of occurrences is measured using minimum convex polygon to capture all known patches of ecosystem extent. And then we have area of occupancy, which counts the number of 10 by 10 grid cells that are contained within the 10 by 10 grid cells. So the catch with applying this criteria is that we need to have evidence of ongoing decline. It can either be spatial or functional decline, but there has to be that evidence. If there's no evidence, then we cannot apply this criteria. And then for terms of the data needed to apply this criteria, basically need the ecosystem map. as well as either land cover change or data on functional changes of an ecosystem. And then the other two criteria that focuses on the functional changes of an ecosystem is criterion C and D. These criteria are similar in terms of how the approach that is being applied to assess, but they are different in the sense that criterion C focuses on environmental degradation, while criterion D focuses on biotic disruption, right? So to apply this criteria, you need to have special explicit data that represent ecosystem extent that is affected by degradation. And you also need to have suitable indicators to measure the severity of the degradation. So this is one of the most challenging criteria to apply because of data limitations as well as knowledge limitations. And we hope that everyone, people within, expert within this room will help us in assessing or applying this criteria. In terms of the grassland biome, the assessment for the grassland biome relied heavily on criterion A and B, and that is because of we have a good land cover change data set that allowed us to measure changes in ecosystem extent. However, I mean, yeah, and this gave us, or it fueled us to be able to produce accurate and reliable assessment, but we were not able to comprehensively assess functional criteria under criterion C and D due to data limitation as well as knowledge gaps. So this, as I mentioned, the assessment under for the grassland biome were largely driven by criterion A and B. So what this also means is that many of the ecosystem types that have been listed as least concerned are likely to be threatened because of we're not able to assess degradation or functional changes well. So, but we are confident in those that have been listed as threatened because of the good land cover data that we do have. Ecosystems that are threatened by both degradation as well as habitat loss, those are likely to be listed as threatened because of they would have been picked up through the spatial assessment. Okay, coming out of the NBA, approximately 60% of the natural habitat in the grassland biome remains in a natural or near natural state. And then 24 of 74 ecosystem types that are found in this biome are threatened, and they cover about 28% of the natural habitat that is remaining. And approximately 5% of the remaining natural habitat is modified, and that was between 1990 and 2024, which makes grassland biome the most impacted biome. The second most impacted biome. And the drivers of this change are largely the croplands with 30%, covering 30%, as well as plantations. Okay, in addressing these data gaps, Sandy, as Wataru mentioned, is leading a regional project called SPEP for short. And this project is aimed at developing the National Spatial Database on Ecosystem Condition and Integrity. And the aim is to develop a database that is fit for purpose and that is for the readiness of ecosystem assessment. We would like to improve the readiness of ecosystem assessment so that the ecosystem that are now downlisted we may be able to uplist, and also to inform biodiversity monitoring as well as reporting. So there are plans to address these data gaps. Sanbe would also like to, or is planning on establishing a committee, Grassland Biome Readiness of Ecosystem Committee. Yeah, so we are inviting you if you are interested. And the purpose of this committee is basically to collaborate in our efforts of mapping and interpreting ecological indicators for the grassland biome. We want to create this database, and we need your input in doing that. The other thing is that we would like the expert within this group to be able to undertake assessment. Obviously, we don't say that the assessment at the national level, but if you are interested, if people are interested and have got the data, they can select one or few ecosystem types that they can assess and uplist. And obviously, Sanbi will make sure that is available to guide and provide all the resources that are needed. And lastly, we also invite you to help us in terms of reviewing our assessment, which is the same approach that the threatened species program is doing. So we want to give credibility to our assessment, and therefore we invite you to help to review our assessment and provide validation. Yeah, please get in touch. Steph, can you please just raise up your hand? Steph is one of the SBEP team that is working on developing this national database. Please get in touch with Steph or myself if you are interested, but we'll greatly appreciate your contribution. Thank you.
Thank you so much. Sorry to be the boogeyman, but thank you very much, Wapale. And we are going to give Nelmarie's, sorry, your time is well up, so we'll save any questions for the discussion. We're going to see if we can get Nelmarie back on video with sound.
Good day. I'm Nelmarie Simon, a rangelands researcher at the Western Cape Department of Agriculture. Today I'm going to tell you about what we found could work or not when trying to rehabilitate old potato production circles in the Luipoltville sand fynbos. The Luipoltville sand fynbos vegetation type, shown in red, is a semi-arid endangered ecosystem with acidic sandy soils situated in the Sandveld region along the west coast of the Western Cape. Less than 45% of its natural vegetation remains. The rest is cultivated lands, of which 27% is under pivot irrigation and used for potato production. You can see all the pivots in this satellite image. As input costs rise and soil diseases proliferate, many of the lands are being abandoned and left to recover through natural succession. Natural recovery of degraded semi-arid land is, however, a very slow process. The Luipoltville sand fynbos is known for wind erosion problems, and years of cultivation and fertilization have drastically altered the soil chemistry, shifting it from acidic with low phosphorus levels to basic with high phosphorus levels. The high phosphorus levels inhibit the establishment of fynbos species. Therefore, some form of active restoration is necessary to ameliorate the soil condition, reduce wind erosion, and initiate the establishment of natural vegetation. The objective of this research was to rehabilitate the abandoned lands to a more semi-natural state that could improve ecosystem services through the reduction of soil phosphorus levels, the establishment of indigenous plant species, and lastly, mitigating the impact of wind erosion. That brings us to the material and methods. The study was done at 3 sites in the Leipoldville sand fynbos. This winter rainfall area receives an average annual rainfall of 263 millimeters. All sites, characterized by deep sandy soils, had been removed from cultivation 5 to 7 years prior to the commencement of this study and was in different states of natural recovery. The first site is near Redlinghuis. It is a flattened dune used for potato production from 1974 until 2012. By 2017, there was minimal permanent plant cover. The phosphorus levels were at 64 mg per kilogram compared to the adjacent veld's 4 mg per kilogram, and the organic carbon levels were very low at about 0.1%. The second site near Sandberg had lain fallow since 2010. It had a good perennial plant cover, but it was mainly cynodon-dactylon. The soil phosphorus levels were 36 mg compared to the veld's 7 mg per kilogram, and the organic carbon was at almost 0.2%. The third site was located near Elands Bay. This site was last cultivated in 2010. The site had a good cover of Erharta calycina, but few other species. Its phosphorus levels were at 29 mg per kilogram compared to the 10 mg per kilogram in the adjacent veld, and the organic carbon content was at 0.15%. The same experimental design and layout were implemented at all 3 sites, following a randomized block design with 4 replicates and 7 treatments. In an effort to combat the high phosphorus levels, an initial rye mix was planted in 2017 and 2018. Minimum soil disturbance was done with a tine implement to prepare the soil, and after sowing, the soil was rolled to ensure good seed-soil contact. The mix consisted of cereal rye, lupins, and Cerridella. These species are known to withdraw phosphorus from the soil. Due to the drought in 2017, which resulted in almost zero germination, the planting was repeated in 2018. The plants were cut down and material removed from the land before seed set in an effort to remove the phosphorus from the land. To combat wind erosion, brush packing was done with branches from local Port Jackson trees in 2020, which also provided organic matter. Branches without seed pods were used to prevent the spread of this invasive species. Wind speed can reach 11 meters per second at Redlinghuis and Elands Bay, where signs of wind erosion was the most visible, as seen in these photos. Lastly, cuttings from species indigenous to the area were made and planted in the plots. This was in an effort to increase the species diversity on the land. Plant material was collected in the adjacent Lambert's Bay strandveld, where the natural soil phosphorus levels are higher and the plants are adapted to these phosphorus levels. Seeding with Erharta calycina collected at Elands Bay and Erycephalus racemosa was done in June of 2020, and in June 2021, seeds of Tyronia onobromoides was sown. Erharta calycina were only sown at Sandberg and Redlinghuis, as it is already present at the Elands Bay site. Cuttings of Osteospermum moniliferum, Monoclamus albicans, and Exomus microphylla were planted in June 2021, and then combinations of these 3 treatments and the control made up the 7 treatments in total. Soil samples were collected in all treatments in May 2017 and October 2023 to determine any changes in phosphorus levels, organic carbon, and microbial diversity due to treatments. Survival of the cuttings was monitored until August 2023. Data was analyzed using ANOVA, Fisher's least significant difference test, and principal component analysis. So what did we find after 6 years? By September 2021, the cuttings of all species were well established across all sites. It was up to this stage in the study that I reported on at the 57th GSSA Congress at Aldam, where the subtitle of the poster was, "Don't count your chickens before they hatch." We know that for seedlings and cuttings to survive, they require water and protection from the harsh weather conditions, such as wind and high temperatures. So the question now is, what happened to the chickens? Significantly more cuttings survive at Elands Bay and Sandberg sites than at the Redlinghuis site, with Osteospermum moniliferum being the most successful over all the sites. The hot, dry summer took their toll and many plants died by June 2022. However, at Redlinghuis site, which had the least natural cover, almost all the plants died by August 2023. Here at Redlinghuis, survival was better in the treatments with brush packing. Interestingly, the survival of the cuttings was the least in the initial rye mix and planting treatments at both Sandberg and Elands Bay, especially compared to the planting-only treatment. A possible explanation for this is that the plot was cleared of any existing plants during soil preparation, leaving the soil barren with no natural cover or branches to protect the plants from the harsh environmental conditions. Less than 1% of the Errata seeds sown at Redlinghuis and Sandberg sites had germinated and established by June 2022. At Redlinghuis, all these plants survived in the brush packing treatments, and at Sandberg, more than 50% of the plants survived, which was significantly more than in the treatments without brush packing. Again, showing the importance of soil cover, either with plants or something else like branches on the soil. None of the other species seed established and survived. Due to the drought experienced from 2017 to 2020, the initial rye mix established poorly and had little impact on the soil phosphorus levels. However, the phosphorus levels decreased over time at two of the sites, but it increased significantly at Elands Bay. The Elands Bay site is adjacent to an actively used cropland, and it is possible that the phosphorus could have blown onto the old land. The organic carbon increased over time in all the treatments at the Elands Bay and Sandberg sites. At Redlinghuis, with the lowest natural plant cover on the abandoned land, the organic carbon only increased in the treatments that included brush packing. The branches were partially covered with sand, adding more organic matter to the soil. This emphasizes the importance of brush packing, not only for the protection of the young plants against harsh climatic conditions, but also to improve the soil conditions. At all the sites, there were a significant increase in soil microbial diversity. The first axis of the PCA shows a clear difference between the initial sampling in 2017 to the left of the graph and those done in 21 and 23 respectively to the right of the graph. There was significant more microbial activity at the Sandberg site than the others, probably because it has the best natural plant cover. The improvement over time was the best at the Redlinghuis site, and specifically in the treatments with brush packing, perhaps due to more organic matter in the soil and more plants compared to the other treatments at this site. This increase in the soil microbial diversity is an indication of an overall improvement in the overall soil health. Although brush packing delivered the best results in terms of survival of the planted species and improvement of the soil, as it lowered the impact of wind erosion and provided a better microclimate, we expected it to be even better. A possible reason is that it was relatively small plots of 5 by 5 meters that were more than 10 meters apart and far from natural vegetation. We recommend that for brush packing or any other form of protection against wind erosion, one should start as close as possible to the natural vegetation that can act as the first line of defense against the wind and work over as large an area as possible. If the impact of the wind is under control, the plants will have a better chance of survival. On an abandoned land with a permanent pioneer plant cover, brush packing is not necessarily required, but it will benefit from the sowing or planting of later successional species such as Osteospermum moniliferum and others to improve the biodiversity. Erharta calycina proved to be an excellent pioneer plant, especially when some protection is provided on bare lands. In conclusion, sufficient rainfall and protection from wind erosion are critical for the survival of young plants and the improvement of ecosystem services. Abandoned lands with the least natural cover benefit the most from restoration actions. We've seen activities of birds, hares and other small animals on the lands, especially prominent at the Riedlinge site that was originally rather bare, indicating an improvement in the overall ecosystem health since we've started with our rehabilitation actions. Thank you.
Do you anytime look at the Forbes and their reactions to these different things?
Okay, so my project, I was only looking at the grass species, but my colleague did look at the Forbes, and if I remember, so she didn't look at the predictor, she was looking between if mutualism was beneficial to them or if the competition was higher. And she found that at the scale that we were working at, it was by pure randomness that they were there.
Are there any other questions? Urs?
Yeah, I've got a question for Wataru. In the graphs that you showed, it seemed as though where oh yeah, don't know where I'm talking to. Oh yeah, good. It indicated that you had an overestimation at low density and an underestimation at higher density of cover. Was that correct? Is that correct interpretation?
Yes, that's confusing because I'm not predicting vegetation cover. I predict bare ground. So
All
that
right.
graph I showed is the overestimation of bare ground, which is underestimation of vegetation.
Right. Okay. I had it backwards. But anyway, did you make any adjustments for that in your predictions?
I haven't made any adjustment for now because I still need to compare with field data, so that because I'm not 100% sure my photo interpretation is perfect. I think it's reasonable, but yeah. So I want to look at field data before doing that.
And then you gave some examples where there was not a very good match. There was a fair mismatch. What was did you figure out why there might have been a significant mismatch in the areas that you showed us?
I wanted to ask some experts here what would be the possible reason for that. Obviously, soil could play a role, but the problem is the good soil layer probably is not available for that part of the Karoo. But yeah, so if anyone knows something, I really want to hear about that.
I wonder if it's got anything to do with soil reflectance, that you might have some interference from the soil, different soil types.
Exactly.
Linda, some questions from the Zoom. Cool. Did you hear that? Nelmarie is available. Sue.
Thanks. For my Sanbi biodiversity people and the grassland biome ecosystem classifications, I just wanted to find out, I may have missed it in the beginning. Are you just using the SANLEC data sets to compare range expansion or contraction, or is there more detailed data? Because we know from Maputoland it's completely wrong. And that there's this, it's really difficult to discern between a woody encroached grassland and a grassland. So just how they're dealing with that and their assessments. Apologies, I probably didn't listen in the beginning.
I think it wasn't SANLEC data, I don't think. It was national land cover data that you were working with.
Can we get the definitive answer in the microphone, please?
Can you please repeat that?
Data, we've got, whoops. There's a time series of data sets on the South African land cover. Is that the data that was used for the grassland biome assessment? Yes. Okay, we'll be able to help you with some better information on the Maputoland coastal plain because it's very wrong. It totally gets it shows an increase in grassland when there's actually a decrease, and it's because of the different ways in which those data sets were produced, but it's also this issue of trying to discern a woody encroached grassland from a healthy grassland. It's quite tricky.
Yeah, no, thank you. Because we do welcome fine scale land cover data to supplement our assessment, and that has happened in the fynbos biome, whereby they provided us with additional land cover data, and that helped in uplisting some of the ecosystem types.
Yeah, so we are busy developing a bush encroachment layer to feed into the degradation criteria she spoke about. So that would help discern the true state of those grasslands.
Urs has got another question or comment for Nelmarie.
Can Nelmarie hear us online? Okay.
Yes.
Nelmarie, I was just curious by the fact that you had a high concentration of phosphorus in the soils that had previously been calculated. More often than not, we worried about a low concentration of phosphorus. Was it the fertilization that resulted in that high phosphorus? And then if that was the case, why is it likely to have persisted so long?
Okay, so yes, it's the fertilization. The fynbos is known for very low levels of phosphorus. So that is because of fertilization that it's in the old lands, it's so high. And phosphorus doesn't leach easily. So that's why it's still very high.
Can I ask a question of Nelmarie while we've got her on the line? So I was very interested to see, I mean, first of all, wow, those blasted bare sand patches. It's quite a challenge to rehabilitate those. But what is the actual sort of envisaged final state and also final land use of these once they are rehabilitated?
Well, there's not really, that part is not really used like for livestock, but they do use it. So it is to improve the ecosystem because it's in an endangered veld type. So just to get it a bit better, but also then to provide food for livestock if needed. But yeah, it's just to improve the overall health of the vegetation type. I don't think we'll ever get to what it is in the what do you call it? what it must look like.
The sort
we
of
won't get it there,
original
but
Yeah. But hopefully it's better than what it was. Well, it definitely is better than what it was.
So now we have to feel guilty about eating beef and the potatoes with it. Has anybody got a study on broccoli? Are there any further questions for our speakers? It seems maybe the beef and potatoes is calling us. Yeah, thanks very much to all the speakers for a very stimulating session and for the audience for the active participations.